Hello everybody, I'm Aldo Pastore, postdoctoral researcher at Scuola Normale Superiore di Pisa and Fondazione Pisana per la Scienza. Today, I'm going to introduce you to the work that I will present at the conference in Lyon. The precision medicine in glioblastoma is still not very efficient. Indeed, in the context of this disease, most of the patients are treated with the same chemotherapy, named temozolomide. Despite of that, recently, functional assays have shown their potential in drug testing, offering a way to enhance patient stratification. Among the functional assays, in our work we will focus on a very promising one, the Fluorescence Lifetime Imaging Microscopy, which is an imaging technique to detect metabolic changes in a tumor sample. Importantly, these changes are directly correlated with early treatment response. However, as many functional assays, it lacks of retrospective data, limiting its clinical translation. Our aim, therefore, is to solidify the clinical significance of this imaging technique. To do this, we exploited both a cohort of glioblastoma samples collected in our laboratory and an external cohort of glioblastoma patients with matched clinical data. And in particular, among clinical variables, we leveraged progression-free interval as an approximation of drug response. Finally, to further validate our approach, we developed a predictive model based on the film-derived information. Our cohort of glioblastoma explants is constituted by 33 samples, treated with the temozolomide. The samples have been consequently labeled as responsive or non-responsive based on the presence or absence after the TMZ treatment of metabolic changes identified by the film. Using the RNA-seq data of the glioblastoma explants, we thus performed a differential expression analysis, comparing these two groups of samples. In this way, we obtained 244 differential expressed genes, representing the genes involved in the TMZ response based on the film classification. To obviate to the lack of retrospective data, we exploited the glioblastoma dataset of the Cancer Genome Atlas. In particular, we selected the TMZ-treated patients with matched RNA-seq and clinical data. Again, we defined two groups, named long and short PFI, so this time we used the progression-free interval of the patient to define the groups. Finally, we performed a similar differential expression analysis, finding 1090 differential expressed genes, representing the genes involved in the disease containment. We then intersected the two sets of differentially expressed genes, finding a non-null set of intersection. This subset of differentially expressed genes therefore represented the ones indicated by the film that also carried clinical information about the disease containment. Among them, using a discriminant analysis, we selected the ones with the highest capacity to discriminate between responsive and non-responsive samples. And here we can see the expression levels of the 6 selected genes. Interestingly, all the genes are consistently overexpressed over the two datasets in the worst prognosis condition, so non-responsive and short progression-free interval. Finally, to add a stronger clinical value to the identified set of genes, we used them to build a predictive model. With this aim, we selected a new dataset of glioblastoma patients and extracted the expression levels of the identified genes and the progression-free interval values of the patients. Thus, with the leave one patient out approach, we train a Cox regression. The model links the transcriptomic information to the progression-free interval in order to stratify the patients in two risk classes, high and low risk. As you can see from the graph, the Kaplan-Meier curves of the two groups are highly separated, proving the internal validity of the model. We further tested the model with unseen patients in order to verify the model external validity. The results are summarized by the Kaplan-Meiers on the left. The curves are still significantly well separated, implying that also the external validity of the model is confirmed. To better understand how well the model performed, we compared its performance with the one obtained by using the MGMT methylation status, a well-established prognostic factor. The performance results very similar, supporting the capability of the film technique in extracting clinically relevant information. So, to summarize, using the film classification, we derived a genes panel with predictive value. This establishes a strong link between the dysfunctional assay and clinical outcomes in glioblastoma. The integration of the identified genes into a machine learning model has enabled the stratification of patients into two risk classes, with a performance comparable to a well-established prognostic factor. We highlight that our bioinformatics approach could be tailored to validate other screening tests that cannot be directly investigated with a retrospective data. So, thanks a lot and hope to see you soon in Lyon.